Okay, hello everybody. This is Julian from Straight from a Scientist. We have Miguel here again, back by popular demand, speaking about nutritional neuroscience and the microbiome. He recently published a review article that is excellent. We will link it to the show notes. Yeah, we're here to talk about his new review article and his updates on his everyday research. So, welcome, Miguel.
Hi, thank you for having me again.
Yeah, of course. People loved your last episode, so we got some requests to have you on again, and we're obviously glad to talk to you again. So, anyway, thank you for coming on again.
Thank you. Thanks very much.
So, tell us a little bit about this paper that you recently published a couple weeks ago, right? Or about a month or two ago?
Yeah, it's been a little bit longer than that. It was kind of,
Okay.
yeah, it was kind of April time, late April. Yeah, but it's done really well. It's nearly getting to 10,000 reads, which is amazing, and I'm really pleased. I've been, like the editor has been all over me asking me to do more things, and
Write more, write more. Yeah.
yes, MDPI has featured it on their homepage, and yeah, I think it's done really well. I'm very pleased because when I wrote it, I thought, shit, this is like 40 pages or something. Nobody's going to read it, and but I've had loads of interest from all over the world, so it's been very cool. So what I wanted to do really was to have a look at this area from a clinical perspective, because there's a lot that is written that is based on either basic science or animal models. And of course, we know a lot of these things about the communication between the gut and the brain and so on because of the animal models and the basic science. But when you try to apply it clinically, then you're really making too many assumptions. And when you want to be a little bit more sophisticated with your approaches, then assumptions based on mice and test tubes are just a big jump, basically. So I over the months I collected a lot of literature, and I reviewed it, and I it was kind of like a narrative review as opposed to a systematic review. So I've looked at things that actually have got an implication in clinical practice that is tangible and that is practical and that is easy to understand. So I did a lot of the kind of analysis in advance, and I chose a few papers. Well, in fact, I think my reference list is about 200 papers, so quite a few papers.
Just 200.
Yeah, just 200. But I mostly is clinical data. So there's always a few papers that you need to quote that are based on stuff that has happened in a lab, but mostly it's about clinical stuff, and the kind of markers that I find most useful in clinical practice as well when I'm looking at the gut. Some of them are obvious because there are things that happen in the gut and stay in the gut and kind of have been used for many years for the gut, but I wanted to give them a little bit of a twist, and find out if they are connected with the brain in any way. So that's the kind of gist, really, about the paper.
Yeah, I wanted to ask you before we get too deep, some people don't understand what makes up the gut. So the gut is not just the stomach, right? The gut is the stomach, large intestine, small intestine.
Yeah, well, the gut, if you want to look at the gut really as a whole, it's from your mouth to your anus, basically.
Okay, so it's the
So
entire
that's,
tract from your mouth to your anus.
yeah, because you'll have different bacteria and different secretions, different kind of juices going on from one end to the other, and they'll play a role in immunity, in communication between different organs, in how you process foods, in how whether you are more likely to put on weight or less likely to put on weight, or more likely to feel happy or less likely to feel happy, or all this kind of stuff. And so it's if you look at where the concentration of bacteria are, the stomach, you have some bacteria in there, but it's like the upside down in Stranger Things. It's just very hostile.
Yeah.
So nobody wants to live in the upside down. It's kind of like totally messed up.
There's a lot of
So
acid and yeah.
a lot of acid, a lot of churning. So it's chemical and physical. So it's a lot. It's like like an earthquake, because the stomach actually is kind of making movements to churn the food into baby food. And then there's the extreme acidity. So only a few bugs can actually survive the environment to actually settle in the stomach, and they are not ideal bugs. So if anybody knows about ulcers, for example, like peptic ulcers and stomach ulcers, there's a specific bug that is called H. pylori, that actually settles in the stomach. Is one of the few bugs that is able to stay in the stomach and make a colony in the stomach, and it does that because it grabs the lining of the stomach with like this kind of hook, and by doing that, it actually perforates the stomach, so it actually helps the making of an ulcer in the stomach.
Wow.
So
I didn't know that at all, actually. The only thing I've heard about H. pylori is that I had a friend that was tired for months and had no energy, and finally got tested for, stomach bugs, and like a stool test, and she found out that she had H. pylori, and when they fixed it, she said everything went back to normal, and so that's crazy. Yeah, I didn't know that it actually causes stomach ulcers. That's very interesting.
Yeah, it is related to ulcerations. So that's one extreme. There are a few other bugs that can kind of just stay in the gut, in the stomach for a few days, but nothing really stays in the stomach for a long time, just because it's just so hostile. And then you've got the small intestine, and you do have some bacteria that stay in the small intestine, but the most chunk of the bacteria are actually in the colon. So
okay.
So you've got the large intestine is the environment, the kind of like the habitat for most of the bacteria that we talk about as gut flora or microbiota, or some people call it the microbiome, although the ideal word is microbiota, but it sounds less sexy. Microbiome sounds a bit more sexy. Microbiota. So microbiome is the microbiota, so the bugs and their genes and the kind of like connections between them, and microbiota is just the actual bugs, the actual microorganisms. So it's not only just bacteria, it's also yeasts, it's parasites, it's anything that's kind of small and alive inside you. That's the microbiota.
Very interesting. Now, my question to you now is that how, if you take a probiotic supplement or drink or eat some kefir or drink some kombucha, how much of that is going to reach the large intestine without being killed by the stomach and, lost on the way?
Yeah, so that's a good question. So there has been studies on viability of the probiotics, and it really depends on the dose that you take. Kefir and kombucha are actually quite concentrated, so when you drink a glass of kefir that you've made at home, whether it's water kefir or milk kefir, and I referred to them in the previous podcast that I did with you guys, so if you're linking up that one, then whoever didn't listen to that has the opportunity to go back and listen to me talking about the kefir and the kombucha. But the kefir and kombucha, probably the probiotic making people will hate me when I say that they're probably a lot better than any probiotic in the market, just because in terms of the spread and the quantity of the bugs that you get in those products that you can make at home quite cheaply or buy in the supermarket, like in Eastern Europe, you buy kefir everywhere. You can buy it in Poland, in Ukraine, in Russia for like a few pennies.
It's cheap,
but
yeah.
If you go to California, you pay like $10 for a bottle of organic kefir, because it's kind of like a trendy food. Or exactly. London is the same. So it's but it's very concentrated, and the ideal way to do this is by making it at home from grains. So from the SCOBY, which is SCOBY stands for symbiotic colony of bacteria and yeast, and it's basically it does what it says on the tin. It's just it's like a little kind of a mini sponges. It's like a clump of bacteria and yeast together, and they ferment the sugars in kombucha. Is the sugar that you add to the tea, and the actual tea as well. The catechins in the tea are fermented and they are released into the water in with other names and other kind of formulations. And in the kefir, it's very straightforward. The lactose is eaten up by the bugs, and so it's not sweet. It's kind of like tarty. It's a bit like sheep's yogurt or goat's yogurt, that kind of like a slightly kind of aftertaste. And it's got billions of the beneficial bacteria. In a paper that I'm not able to quote exactly now, I should do. Once you ask me another question, I'll have a quick look at my computer. This viability study done on water kefir with sucrose, basically with sugar, that found 57 lactic acid bacteria types in just regular homemade water kefir. That's 57 types. So imagine going to a shop, you go to your Whole Foods or Planet Organic or your pharmacy or something, and you ask for a probiotic, and the more bugs and the more variety and the higher the numbers, the more it costs. This would cost you like 200 quid or $200 to buy, a pot of probiotics that would give you that kind of quantity and spread of bugs.
Yeah, I've seen actually Whole Foods. I've seen one that had, I think, 16 strains. They were like 16 strains, 5 billion, and it's at $40, which is
exactly.
Expensive.
So it's quite expensive.
And I go to Whole Foods, or I go to Mercadona right here, and it's like €1 for the big thing of kefir. So definitely a lot cheaper. Yeah. And so do you make your own kefir at home? I remember last time we talked about you making your own kombucha.
So I make my own. I'm not making the milk one at the moment. I'm making some water one only, just because I find it more refreshing for the summer, and I tend to do the milk one in the winter. So,
it's the same as
the same
kombucha.
thing.
You buy the SCOBY and
you buy the SCOBY. You buy the water kefir SCOBY looks a bit like crushed ice, as opposed to the milk one, which is kind of like sponges, like little kind of like clumps of wool or something. Like
so two different SCOBYs.
Two different SCOBYs, different SCOBYs, and they are vaguely related, but they are actually two different organisms.
Very interesting. Yeah, I did not know that. I've never heard of water kefir. I guess the one that I buy is a milk kefir, because it's more like a very light Greek yogurt. It's very like a watery yogurt more than anything.
Sure. And that's now coming. So milk kefir was kind of... It was kind of esoteric as well a few years ago. It's kind of things that hippie people drink, and like vegans and so on, and and like made with coconut milk and whatever. And it's just become very mainstream in the UK now, and in loads of places in the US as well.
Which is great, I think. I mean.
Yeah, and the water kefir is coming now. There's there's a few supermarkets in the UK that are stocking the water kefir now, so you don't need to go to a super sophisticated shop to buy that.
Yeah, actually, I'm gonna try the water kefir. I want to try to start making it. In the US, kombucha is huge. In Spain, I have not seen kombucha. It's very hard to find. I think it's it's on its way, but I can find kefir, whereas in the US it's very hard to find kefir.
Yeah.
So I guess it just has to do with the culture and
Yeah, of course. Yeah. So if you have an idea of the numbers in the jejunum and the ileum in the small intestine, you have about tens of thousands to hundreds of tens of thousands of bacteria, mostly lactobacilli, because they are quite hardy. If you've ever tried and break lactobacilli to actually extract the DNA, it's like metal. Is one of those that actually you need to take with a pinch of salt when you get the results from a stool test, because it tends to be a little bit unreliable. And that's the case if somebody has been taking probiotics and they do and typically it's lactobacillus blah blah blah, like whatever. It tends to be lactobacillus and bifidobacteria. Particularly the lactobacillus are so hard to break that if it comes as very little lactobacillus, and that the person has been taking a high strength probiotic, that the test has failed to actually break the DNA properly. And this is widely reported in literature, and people who who do this kind of thing all the time, scientists who do this kind of thing all the time, they know that lactobacillus can be underreported just because they are difficult to break down. But the small intestine is mainly inhabited by lactobacilli, some coliform bacteria, streptococci, some bacteroides, some fusobacteria, some bifidobacteria, and between the thousands and the tens of thousands, possibly 100,000, like not that much more than that in quantities. Whereas you have exponentially bigger numbers in the colon, and you have also a lot more bacteria and other organisms like yeast and protozoa as well. So you go into the millions to billions and trillions, basically. So you can basically go into trillions of colony-forming units of things like, bacteroides and lactobacilli, smaller numbers of proteobacteria, which are normally about 2-3% of the total. But you also have yeasts from the same family that will make beer, so the Saccharomyces family that will make beer, and you have protozoa as well. So you have this kind of fluffy things with a hook that allows them to travel around the gut. And most of them, like the blastocystis hominis, for example, is a common infection. It's considered a pseudoparasite. Most people have it. You can pick it up, and then it will go in the next time that you have a stool test. It's gone. The Antoamoeba, so you have some amoebas as well. The Antoamoeba fragilis is another one that is kind of in a lot of people's guts. And normally, clinically, I wouldn't do much about those unless there were loads of symptoms that you could relate to those, and you had a clear understanding that it is those because you've done a few tests and they always come back. So they tend to be innocuous. It's actually there's even a theory that people are kind of considering at the moment in terms of do we need those parasites for health as well? Because they kind of stimulate our immune system. So maybe if our immune system is stimulated inside against a parasite, it may not trying to fight itself. So it's probably going to result in less autoimmunity, for example, because that localized immunity in the gut that is very powerful. About 80% of the immune system is in the gut. So if you keep it entertained with a few little parasites floating about, it's not going to have any ideas to go and attack, the skin or the eyes or the heart or whatever and cause an autoimmune disorder.
Very
so there's
interesting.
this kind of a medical hypothesis going on about how, people who have got parasites and potentially more of a dysbiotic, more of a deranged kind of a gut balance may be at least a little bit protected from that autoimmunity. It's all about anyway.
Yeah, now I have about 7 questions for you after that.
Sure, that's fine.
The first one I was going to ask you about is how reliable are stool tests in... Oh,
sorry.
No, it's okay. You want some food?
Yeah.
How reliable are stool tests when you're trying to figure out what's going on in somebody's microbiome or microbiota? Because I remember one time I went to Mexico for a couple weeks and I came back and I was feeling sick, and I remember going to get some stool tests because I thought I had some sort of gut bug. And the lady told me, well, we have to do 3 of them because with 1 we have 60% chance of finding out, and then 2 we get 75, and 3 we have like a 90% chance of finding what we're looking for. Yeah. So do you agree with that? Is that
Yeah, so that's common practice in parasitology. So if you're talking about parasitology, first, the first part of your question, so how good stool tests are, it depends what you're looking for. But if you're looking at parasitology, they're pretty good because you'll have various methods to identify the parasites, whether it's a kind of like a mapping tool. So you've got a catalog of most of the bugs about in various guts, from records in the past, and you try and map the DNA from those bugs to to a database and try to identify them that way. So by PCR, by this kind of technique that it's kind of DNA identification. The more old school way of doing it is by looking under a microscope and actually try to identify whether there are eggs of the parasite or whether the parasite is actually visible with the naked eye. It could be a worm. So those are pretty easy to identify. So they are pretty good for this kind of stuff. In terms of for the bacteria, there are different ways of doing stool tests. So there are still people doing cultures. Cultures are pretty old school. So if you haven't got anything else, then you might as well do a culture. That's the only thing that is available. But the most reliable way of testing a gut is by doing, again, this kind of same technique as with the parasites, a DNA identification. So it's almost like the most wanted. You've got the most wanted criminals, and you've got a picture of them, and you know exactly who you're looking for, and you're trying to map the faces of these guys in the gut against the faces of the most wanted, and with a lot more precision, obviously. So you know exactly that the streptococci are streptococci, and the eubacteria are eubacteria, and the clostridia are clostridia. So, and to the point that you can identify the strain. So not only the genus and obviously the phyla, the genus, all the way down the hierarchy, all the way to the bottom to the strain. And that is useful in the sense that you actually break down the numbers. So you could compare your gut and your gut profile with the gut of a healthy person, of healthy cohort that has been studied to have, a good diet and healthy kind of, good health and so on. So you've got a comparison between somebody who is healthy and hasn't got any gut issues or any other, issues like diabetes or cardiovascular disease and all this kind of stuff. And that's actually very useful because it gives you a benchmark, So this is your gut compared to this healthy cohort. And a lot of the tests that are available now, either via spin-offs from universities, like the PREDICT study that was the old Map My Gut that I used to be involved in. That was a spin-off from King's College and Team Spectre. There is also Eubiome that is available.
I've heard about Eubiome, yeah.
Yeah, so they all have this concept. There's MyMicroZoo as well, which is in Netherlands operation. They all have this concept of a healthy gut that is based on either their own data. So Map My Gut or the PREDICT study now, which is a full randomized controlled trial taking place as we speak now, led by Tim Spector as the lead investigator. That's looking at the database of 14,000 people that he's got as part of the twin study, the kind of data he's been collecting for the last 30 years. And within that, there's a subset of healthy guts that he's identified. So he's comparing kind of an unhealthy gut or your profile with loads of people with this amalgamated data. So it gives you a reassurance that it's not just a small cohort, it's not a small group. When you have people, I don't know, whoever, company, blah, 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 selling you a stool test, and the cohort they are comparing to is 20 people in Mexico or something, exactly. Or, you need to wonder how reliable that's going to be. That's the only thing.
Yeah, that leads me to another question. How different are the guts of different people, across countries, across ages, across sexes, everything? Is there a big difference between everybody's microbiome?
Yeah, there is, and if
so that complicates a lot the process of trying to compare against a good control, right? Because you don't know what is a good control.
Exactly, and I think the ecological validity or the context becomes a lot more important in this kind of case, because if you're in Sweden and you're comparing your gut with somebody who's Chinese, there's so many environmental factors that you cannot control. So in Sweden, you'll have a lifestyle that, there's so many things we don't even know about. Maybe even the exposure to light is going to have an impact on the type of bacteria that are in your gut. If you compare that with somebody in Southeast Asia, that may have a lot more hours of sun exposure, and because of the hours of sun exposure, they may be happier because they may have, more different biochemicals in the brain going on. Neurotransmitters are floating that are not in Sweden or in Finland, in the middle of the winter. So that may actually have an impact on the gut.
Exactly.
And we know that. Lifestyle cycles from childhood and birthing process, if you're born vaginally, that already carries like an insurance kind of for you, because it gives you a lot of bacteria all at once. Your body's like a sponge. Your immune system is looking for cues as to what is good, what is bad, and you get exposed to this massive amount of bacteria that's in the vagina, and it's
breastfeeding too, right?
And breastfeeding as well. So it's the bacteria in the milk. But also the bacteria around the nipple and the kind of like the dirt, because even like if you're clean, you're still dirty, you still got bacteria floating around you, and that's good. That's good for the baby. But also whether you exercise or you don't exercise, whether you're stressed, whether you're very sedentary, your diet obviously has got a huge impact. Pharmaceuticals, if you take PPIs, they inhibit these kind of antacids and things for indigestion and so on, this Gaviscon and all this kind of stuff that people take, that inhibits your stomach acid. So it's all good. You're not going to feel like burning coming up your throat, but at the same time, it's going to completely derange the process of digestion, and it's going to change your bacterial profile, and maybe making it easier for bacteria that really shouldn't stay in the stomach or the small intestine to take a hold in there, because suddenly it's not so inhospitable. It becomes a little bit better to live in there. So they think, oh, well, I'm not going to bother going all the way to the colon. I might as well stay here.
Very interesting.
So there's loads of factors to take into account. It makes it very complicated, but it doesn't need to be super complex, It can.
But it is very. I think it's one of the most complex things to study, because you're not studying the human body. You're studying an external organism that affects the human body, and not only one. You're studying thousands or millions or billions, right, of types. Now, this is very interesting. You talked about C-section versus vaginal birth and breastfeeding. I wanted to talk, we were talking about this a little bit before the podcast about sex and the microbiome, and you had some interesting thoughts about this. So let's bring up that conversation. So we talked a little bit. So you said that it can be good and bad, of course, depending, to have multiple sexual partners, and depending on your sexual partners, you will, transfer bacterias and parasites and all that stuff.
Yeah. So
have there been any studies on that?
Yeah, a few, and I know about this just because I've been looking at them, because I'm writing a paper not just on sex. I would love to write a paper on sex and and the microbiome. That would be fascinating. In fact, I can see myself being a bit of a niche scientist doing sexual studies on the microbiome. That would be fabulous. Yeah, but there's loads of them on the pathogenic end, so basically on the nasty end. So you get gonorrhea, or you get chlamydia, or you get, so that's microbiome as well. So your gonorrhea is a bacterium that wants to come and live inside you, but it's not likely to live in your gut. It's more likely to live either in your anus or your vagina or your throat. So, and same with chlamydia. So there are, we've got microbes all over the body, on the skin, on the eyes, on anything, anywhere that's a little bit moist is going to have microbes. So there is however, the positive end. I was just looking at a couple of papers, and one of them actually stood out, and is published in Cell in 2015 by some guys from the University of Texas, Austin. And it talks about the beneficial sexually transmitted infections. So it actually looks at STIs as a beneficial way to get a better diversity of bacteria. And it quotes, it's an understudied phenomenon with important implications for the evolution and cooperation of the host reproductive behavior. And it basically goes on to say that it's natural to have different partners. So I'm not saying that people should be totally promiscuous and go out and hey, it's good for my microbiome to have 10 different people in one night. It's not all about that. But what it's basically saying is that there is an advantage, and it's part of possibly how we have acquired our current microbiome. And some people are obviously in monogamous relationships. A lot of people are not. Other organisms like insects, for example, it talks about mosquitoes and fungi and aphids, and it's a very interesting paper. And I would like to make a little bit of a point in my next paper about this. Because I'm writing about lifestyle impact on the microbiome. So I'm looking at different aspects of lifestyle, and one of them is sexual behavior. Another one is profession. So if you are a dentist, for example, and you're inhaling various chemicals in your practice, how is that affecting your microbiome? So I'd like to look at kind of various things like that. Because when I'm looking at as a clinician. So if you came to see me and you said to me, one of the things that I like pizza on a Thursday, and on a Friday, I go out to a sex party and I have sex with 10 people. Exactly. Well, both will have an impact on your health. It may be positive, it may be negative, but you cannot just ignore it. And I think whenever it comes to sex, we become either giggly or just ignore it. And I would not like to ignore it. I think it's an important part of lifestyle medicine.
Yeah, and I definitely agree with you. The paper that I read about the microbiome stated exactly the same as you, that you share a lot, especially if you are with a partner for a long period of time. You're sharing a lot of the same bacteria, and not just bacteria, but microbiota with your partner.
Yeah.
And it's not just sex, it's kissing, it's oral sex. I mean, it's a wide, just living together can make a big difference too, right? So
definitely,
very
and I've got 2 dogs and a cat, so I'm sure I've got various different bits and bobs from them as well. And in fact, I found out when I had one of my stool tests a couple of years ago, I had a couple of types of bacteria that are normally seen in rabbits. And where I live, I'm in between London and Cambridge, in quite a rural area where there's loads of rabbits in the fields around here, and my dogs like to eat rabbit poo. And then I kissed my dogs, not like I kissed my dog and I liked it, but I kind of I give them a cuddle and a kiss, and of course they are carrying all sorts of bacteria in their mouths and in their faces, and so I'm getting mice bacteria, rabbit bacteria in my gut as well.
Very interesting. Do you know much about toxoplasmosa? Because I've heard a lot about that related to cats and how it has a strong effect on rats, but we don't really know a lot about humans. And so the toxoplasmosis can be dangerous, particularly for females, and pregnant women. So and it can kind of mimic glandular fever. So it can have the same kind of symptoms as glandular fever. So kind of like swollen glands and extreme tiredness. There's some kind of theories saying that if you get that, then you're more likely to get things like chronic fatigue or ME, because it's a hyperactivation of the vagus nerve and the hypothalamus pituitary adrenal axis, or the HPA axis. So your nervous system gets hyperactivated, and because of that hyperactivation, it gets very tired and very sensitized to stress, so you become less resilient to stress. So anything stressful makes you go funny, and you cannot really deal with it. And that's one of the kind of traits of ME, that any tiny little thing that you just think, oh, fine, you don't even pay attention to it. To somebody who has got that rigidity or has lost the flexibility in their nervous system, it becomes very difficult to manage. And there's a link between. So I'm not an expert in toxoplasmosis. I know a little bit about it, mostly theoretical stuff. I haven't had any first-hand experience with it, but definitely
there is talking about various different bugs coming into your gut and how that they can have an impact on the not only in the balance of bacteria and other organisms in your gut, but also on the integrity of the gut. And the guys from Ireland, the Dynan and Cryan team, that just produce papers as they, as I make donuts at home, these guys just publish in all of the top journals in the world. I changed my underwear, and by the time I've done that, they've produced 3 papers. It's just incredible.
That's crazy.
And they talk about a leaky gut theory of depression, so and mental health. So Dynan is a psychiatrist, and Cryan is a neuroanatomist, and they have this theory that changes in the gut wall permeability, or what it has been called in more alternative circles for a long time, leaky gut. So this is kind of like something that was a bit like woolly, and nobody really know whether it was a real entity or not, and it's showing to be a real entity. Intestinal permeability is a reality. It results in translocation of bacteria, so bacteria move from areas of the gut that they normally don't tend to populate, so they go to other areas, and that changes the whole ecosystem. So it's almost like you've got a forest with bears everywhere, and the bears only choose to colonize one spot of the forest, and you have the squirrels go and colonize another spot of the forest, and they don't kind of interact between them. So it becomes more compartmentalized, the gut, and it loses the natural ecosystem. And then what happens is that the vagus nerve carries a message to the amygdala that is quite strong from the gut in this kind of state, that is inflammatory. It's normally triggered by this thing called LPS, which is a lipopolysaccharide that is like a toxic compound that the bacteria secrete into the gut, and that basically makes the immune system react. So this reaction from the immune system is communicated to the brain, and the brain thinks that you're under attack all the time. So it kind of like triggers a very similar situation as if you were in a fight or flight response. But the message that is telling you that you may need to fight or flee is not coming from the outside. It's not a lion that you're seeing, or it's not a situation that you're imagining. It's actually your bacteria playing tricks with you. And those kind of substances that are being secreted into the gut, and the local immune system in the gut is picking up and transmitting as a radio kind of wave through the vagus nerve and into the amygdala, is just being picked up by that kind of like sensor of stress in the middle of the brain, thinking that you're under attack, and then you basically get stressed out and potentially depressed as well.
Very interesting. So there's a strong connection, is what you're saying, between mental health and stress, depression, all that, and the gut, right? I mean, this is something we're starting to see over the past couple of years, but it's starting to gain a strong place. I wanted to ask you earlier, how plastic is the microbiome? How easy is it to change your microbiome? Let's say you think you just took antibiotics because you got sick and you feel, kind of depressed, tired. How easy is it to replenish and to change and to kind of revitalize your microbiome? And is it possible?
It is possible, and there's been various different theories. Obviously, this is a very emotional subject as well in terms of breastfeeding, for example, because you mentioned it before, and there's been this kind of a movement pro breastfeeding, which I find is great. It's the natural way, so you need to breastfeed if you can. But what scientists are actually finding out is that if you have a baby that's breastfed and you compare it to a baby that's not been breastfed past 36 months, so basically 3 years, the baby that's not breastfed develops other ways to pick up bacteria from the environment. So the baby becomes more savvy at picking up that plasticity that hasn't been naturally fed into him. So if you have been exposed to your mom's nipple and to the kind of nice dirt around the nipple and the milk and so on, and you can, sorry, dog alert.
It's okay.
So if you've been exposed to that kind of situation and you've been able to create a more balanced microbiome as a baby, that's great. But compared to another baby that's not had that advantage, that other baby has developed other mechanisms to pick that up.
So and, they catch up, is what you're saying.
They catch up. Yeah, they catch up about 3. In the latest evidence of that is a good study on Danish babies. That was actually it's a good size, so 300 babies basically compared, and the effects were the variation was negligible.
Very interesting.
Yeah,
I did not know that. I thought that kind of you just had that deficit throughout your life until I guess you fixed it.
No, you can. There's conflicting evidence on this. So there seems that, you know, as a baby your plasticity is huge, so you can pick it up. You can actually make up for it. But as an adult or as a young adult, and it's probably from about 4 years of age onwards, if you insult your microbiome hugely, like with a course of antibiotics or several courses of antibiotics, that can be a problem. So I think that there's like more of a consensus on that if you take repeated courses of antibiotics, that can take years to repair, as opposed to not having enough bacteria initially and then picking it up later.
So what you're saying is maybe you weren't breastfed, and maybe as you get older you have various sexual partners, you live in, you know, the outdoors, you have dogs and cats. Eventually you're going to catch up to have a strong microbiome. You
can, and I think it's an ongoing journey. It's almost like, you know, it's a flight transcontinental from, you know, London to LA. And it's going to go at different altitudes at different parts of the journey. So at certain points of the journey, you're going to be super good in terms of, you know, all of the anti-inflammatory bacteria, because you've had lots of really nice fiber and red berries and, you know, and hydration, and you've been happy and exposed to the light, and you've been doing loads of sports, and some other parts of the journey you've been stuck in an office and miserable, your diet has been crap, and you've been eating burgers and chips, and you know, and it's had a negative effect on your biome, but in the end it kind of all balances out.
Yeah, and you know, I feel the exact same about my own microbiome journey. You know, it has its ups, it has its downs. Sometimes I have, you know, horrible stomach pains and acid reflux, and other times my stomach is strong and I feel healthy and I have a strong immune system. So
yeah,
very interesting. Yeah. So antibiotics, multiple courses, don't take them. Try right, unless your doctor.
Well, if you need them, you
if
need
you really,
them.
really need it.
It's
you
the
need
kind
it.
of thing as well. And like, you know, if you need them, then what you do is then you work with somebody. You know, you do what you can at home. Like, you know, you drink your kombucha and your kefir, and you have loads of fiber. Once you start repopulating the gut, you have loads of fibrous foods, and you eat a rainbow of different colors. That's one of the things that I was talking about in my paper, you know, a variety of different fruits and vegetables, which is, again, the kind of conclusion from the American Gut Project that looked at about 10,000 people, citizen research, people taking stool samples and contributing to this kind of pool of data. And what the scientists in the group concluded was that if you eat about 30 different plant foods per week, and not just plant foods, so obviously if you're a vegan, that's 30 plant foods and that's it. But if you eat everything, 30 plant foods on top of your meat and fish and eggs and dairy and all the rest of it. So if you eat everything, you've got more of a leeway because you can add your fermented dairy and, and fermented meat, because salami has got, is fermented, and chorizo has got a bit of fermentation, and Parma ham or Serrano ham has got a bit of fermentation. All of those will give different bacteria. So about 30 different foods from plant origin and different colors in a week are ideal to sustain a healthy microbiome in the longer run.
Very interesting.
Yeah.
I wanted to ask you earlier, but I forgot about. So alcohol, most of it is fermented, right? You have your beer and wine, and this comes from fermented fruits, right, and grains. So is there any percentage of the bacteria that you're getting from a beer or a wine? Is any of that good bacteria? Or
so you don't really get in beer. You get some bugs that are mostly yeasty bugs from the Saccharomyces family. So Saccharomyces cerevisiae is the bug that makes beer, so you'll get some of that. But mostly what you get that is interesting with beer and wine is polyphenols. In wine, there's obviously resveratrol and other related substances that the bacteria in your gut will try to break down, and they act as prebiotics. So they act as pseudofibers, let's say, because the bacteria cannot really break them down, but they play with them, and in playing with them, they get excited. And think about your bacteria trying to play football or something with these balls that the wine is putting into your gut, and so they are jumping around, kicking about the balls, and they get all sweaty, and this sweat is short-chain fatty acids that are lovely. They are anti-inflammatory, they repair the gut, they repair this integrity of the gut, so there's less leaky gut. There's all of these things are happening. Now, relying on beer and wine as your sources of prebiotics is probably not realistic. So don't get excited if you're a drinker, because it will be negligible, the amount of effect that you will get. But as part of an overall kind of balance, if you have a few glasses of wine and a few beers, then it's all good. Okay. But as gram per gram, you obviously have got all the much better sources of the prebiotics and the polyphenols than the wine and the lager.
Exactly. Well, that's very interesting. I mean, Spain, you're from Spain, and Spain drinks a lot of red wine.
Yeah.
They eat a lot of cured foods and fermented things, and so I think they also are up there on the life expectancy list, right? They're one of the top 5, if I believe.
Yeah.
So that's very interesting. Yeah. So don't go overload on the wine. You can have a couple glasses. It might be nice and might help you, but
yeah.
Very interesting. Okay, let's see. Do I have any more questions for you? I got one more question for you. What kind of symptoms, or how would someone know that they have a problem with their microbiome? What are the symptoms or the triggers or, the red flags that you notice when somebody has a dysbiosis?
Well, it can be a number of things. So it can be quite insidious, and unless you actually have a test, you may not know, because some people are not so well connected with their gut. So and it's something that can be a bit embarrassing for people to talk about. So as an extreme example, I've had a number of cases where people have they go and have a bowel motion once a week, and they thought it was normal because they have never spoken about anybody about how often they go to the loo. So for them it was normal to go just once a week, and they didn't think it was embarrassing for them to talk about it. So for me, it would be terrible, because I go like 3 times a day. So, once a week, it would be like complete disaster. I would probably explode. So for them, it was fine, and they didn't think there was a problem. for some other people, going to the loo 10 times a day with kind of like watery stool is kind of like what they got used to. So it's obviously not normal, but they have got used to that.
Exactly.
And between those two ends of the spectrum, there's a lot of different shades of gray. So most people feel there's bloating, and the bloating may be triggered by something that you can pinpoint. So you could say, oh, there's bloating when I eat bread, or there's bloating when I eat dairy, or there's bloating when I have a heavy meal. And a heavy meal typically is like, a copious meal with loads of fat and processed carbohydrates and so on. So it could be a number of things. Sometimes it's not just the actual bacteria, but it's the secretions, the juices that help the food get into the baby food status, that make it accessible to the bacteria in the colon. Because bacteria in your colon like to get kind of like baby food. They like to get like a puree, like a soup. And suddenly they get this chunky minestrone soup that's got loads of chunks and kind of hearty bits of carrot and so on. They get a bit overwhelmed and they cannot really eat it. So what's going to happen, there's going to be a lot more fermentation. So you could get the bloating. And so the bloating could be partly because your bacteria get deranged and you end up with a limited profile of bacteria that like to eat the chunky carrot soup, and not so much of the bacteria that really like to eat the watery soup. But also it could be because you need to start looking clinically, you need to start looking higher up. So you need to start looking, what is your pancreas and your gallbladder doing? If you feel uneasy when you have a meal that's got loads of a heavy fat, like loads of cream or loads of cheese, is it that your pancreas is not producing enough lipase, and that and maybe supplementing a digestive enzyme, or eating greens that are like slightly astringent, like rocket and things like that, change slightly the pH of the stomach, and you can start or having some apple cider vinegar. So just slightly changing the pH of the stomach, that can actually help. Sometimes it's the gallbladder, and the gallbladder, if it's struggling and you have a very fatty meal, then the fat is going to land in the stool, and you will see that because your stool is going to stick to the pan. So you flush, and there's always like a remnant in there. So that's like typical situation of steatorrhea, so like fat in the stool. That basically means that you haven't processed the fat, you may have just dumped it. And that will have had an effect on your gut bacteria as well. And if that happens all the time, and you just keep eating like this all the time, and you don't pay attention to it, then you're maybe limiting the diversity of your flora. And diversity is the most important marker that we know of thus far. So we definitely know a little bit about loads of things, but the deepest level of knowledge that we have about all of the markers that I've covered in the paper and is about diversity. And we know that diversity, a wider diversity of bacteria in your gut, and a wider profile of different types of bacteria and different strains of the families that inhabit your gut, that commensal bacteria, is likely to become a prophylactic for you. It's likely to become a protective measure. It's going to protect you from the big names like the Alzheimer's and the cancer and the diabetes and obesity and things like that. So it's long answer, but it can be a range of different things. Typically for me, it's bloating, it's burping after meals, it's gas, mostly gas, cramping. So some people call it IBS when it's not IBS, because obviously IBS is very specific kind of a thing. But when they say, oh, I've got a bit of a dodgy stomach, that kind of, it can be, there's 50 shades of gray for different people as to what it means for them.
Yeah, well, let's end on that note, with the most important thing being diversity. Very interesting. So, drink your kombucha, drink your kefir, eat your Greek yogurt, right? 30 different vegetables a week, and you'll get and multiple sexual partners.
Yeah, no, that would be good. Yeah, a few
you're on
at
the
least.
right path. So very interesting. Yeah, thank you for coming on today, Miguel. It's always a pleasure to have you on.
Thank
so
you.
We'll link
so
your
much.
paper, and we have a little summary of the paper that we have had written for us. and we'll link all that in the show notes. So thanks again for coming on. It's been a pleasure as always. I always learn so much. I need to start listening and writing down again all the cool information that you taught us. So thank you. Yeah, look forward to talking to you again in a couple months.
Thank you so much for having me, Julian.
Okay, bye-bye.